Yeah.
Good morning, everybody. Good morning, everybody. My name is Chris Hart, and on behalf of my co-chairs, Eddie Schwartz and Jos Wenke, I really welcome you to this first musculoskeletal infection research interest group meeting. I'm really pleased to see so many of you here, while there's a stiff competition of other sections. Today we will have a fully packed program with 4 speakers, but also in the beginning we would like you to know a little bit why we have set this up and why the reason is and how we want to proceed. So I want to give the word to Eddie, who all initiated this, and I'm really grateful for that, to give you his thoughts on this topic. Thank you.
Okay. So obviously it's a tremendous honor to kick off the first ever RIG for musculoskeletal infection. How many people know what a RIG is within the ORS, what it means, right? Okay, so research interest groups are blossoming new areas. For example, bone, cartilage, ligament, tendon. They have a quarter of the posters, the oral presentations, everything, but evolution of musculoskeletal and orthopedic research is such that new areas of science emerge, and there's a group within it that is, thank you, a group within it that now needs a platform to have presentations and grow within itself. And so the first step to that is having a research interest group meeting. And so I guess 50 years ago, Spine did that. They went on to become the next step, which is something we'll have to have a discussion about moving forward, which is becoming a section. The difference between a RIG and a section is a section actually has officers that are elected by the section. They have a standing meeting. They have an independent budget from the Orthopaedic Research Society to run their section meetings. But they also got to do a lot of work, and they have to maintain the membership. There's dues associated with being in a section, and the attendance of the meeting. So if they're poorly attended, or they're running deficits for the meetings, then the sections are dissolved. So at this point, I'm pleased to see that there's so many people have made this a priority in the busy schedule. As an example, Jeff Richards, who was one of the organizers of the meeting, ran into a conflict and he won't be joining us. So that is one of the issues. And then I'll now turn it over to Josh Wenke, one of the chairs, to talk about moving this forward.
So thank you, Eddie. So Eddie it was his idea and put a lot of the energy and networking through to get permission for this. So we're having this talk at 11:15, and there's a lot of conflicts. Moving forward, the other time is 6 a.m. So then we're going to have 6 a.m. issues, not 11:15 issues. So anybody in here would prefer it at 6 a.m. In the future? Okay, so I think we're going to.
It's good.
Yes.
We have a musculoskeletal infection priority.
No early mornings. So that was one issue, and then as you know, infection is a broad topic, and so what we thought we'd do is break it down into the 3 big things: prevention, detection, and then treatment. And so this year the theme for the most part is going to be prevention and understanding the host immunity and the microbiome and how that plays a role in infection. So with that, ready to introduce the first speaker.
Josh, it's my pleasure to announce our first speaker, Professor Hamish Simpson, who is affiliated to the University of Edinburgh in the UK. Hamish is a professor of orthopedic and traumatology, specializing in limb reconstruction, musculoskeletal infection, and pediatric deformity. On the top of that, he's also the editor of the Bone and Joint Research Journal, and his research interests are in repair of musculoskeletal tissue infections and impaired bone healing. Hamish, if you would please come to the stand and give us your insight. Thank you.
Well, thank you very much for the invitation, and congratulations to the three wise men sitting here who've actually managed to actually get this rig off the ground. It's a great achievement, and really so good to see that infection is getting a better place in the ORS program. So first of all, in terms of okay, great, thank you. In terms of disclosures, I think the only one to mention is we do get some research materials from biocomposites, but we don't actually get any kind of royalties or anything else. It's just a few bits of things, but that's the only thing relevant for the material in this talk. So the title I was given was Gap Areas for Joint Infection and Fracture-Related Infection. I'd like to say straight off, when I was thinking about this, I mean, it's just a massive talk. So I would say that this is a very personal view in terms of things that I would particularly like to know in terms of how with treating patients. Clearly, there's a lot of other things, and other people would include other things in this talk. So in terms of what I've broken it down to, the common clinical areas of patients that come through my clinic, and look at patients with septic arthritis, patients with infected implants, chronic osteomyelitis infected non-union. And what I'm going to try and do here is, it's a bit kind of glary here, but is to try and populate this here, which has got, says common modes of failure. This is kind of a surgical treatment, and this is medical treatment for each of these topics here, and as we go through. So septic arthritis, I think one of the amazing things about infection in the musculoskeletal system is the different ways that it presents with failure. It doesn't just present, in fact, it rarely is actually overwhelming sepsis is the problem from our point of view. It's actually the kind of failure of the musculoskeletal system in other ways. And in septic arthritis, although people do die from septic arthritis, and people do have persistent local infection, the main problem is early degeneration of the joint. That's the one that we'd actually really like to kind of be able to kind of change. And this is just an example here. This is somebody, a young man, 30-year-old, at presentation on the left, and 6 months after an episode of septic arthritis. This kind of incredibly rapid destruction. You can see here this arthroscopy, his cartilage completely gone. But the worrying thing is that when you look at these patients actually on day one, you can actually see a very healthy looking joint, even though you think you've got rid of infection. And the reason is that the extracellular matrix in these patients is still intact, even though it looks as if something else might have gone. And does this matter? Well, actually, this is just showing, again, data from our unit for the hip joint survival after native septic arthritis, showing how many have kind of failed within 10 years. Over about 2/3 or so have failed from an episode of septic arthritis of their native joint. So questions I had for in terms of treating these is, what's the etiology of the damage, and how can we prevent the chondrocyte death? So this is actually work from one of our PhD students, Reese Clement. This is healthy cartilage here on the left, showing a whole load of green chondrocytes. Each one of these are nice and healthy and alive. 95% when the mouse here has been injected with just PBS. On the other hand, this one, after 48 hours of injection of a Staph aureus, which has an alpha toxin, the normal alpha toxin within it, you've got 2.7% alive. So incredibly rapid death of the chondrocytes is created by the Staphylococcus. It did appear from the work that kind of Reese was doing that the main cause of an agent was the alpha toxin. But the other thing we wanted to know was whether or not the host had a response, was affecting it. And so this was the additional panel here, where this Staphylococcus has been genetically modified to have exactly the same characteristics, and it created the same inflammatory response as far as we could see within the limb, but it had no alpha toxin. And you can see with this one, there's only about 20–25% of the chondrocyte death. So it appears there is some kind of death that's probably caused by the host's immune response, but by far the majority is coming from the Staph aureus. So going on to this, in terms of the questions I would kind of want to kind of know in terms of what can we do to minimize this chondrocyte death? Is there a difference in our surgical procedures, aspiration versus washout, in terms of the long-term results? And can we kind of develop a chondroprotective agent that will try and minimize that chondrocyte death. And one thing that a lot of my kind of rheumatology colleagues have kind of raised the question for is they quite like the idea of injecting steroids. To a surgeon, that's often a very kind of worrying kind of thought to actually inject steroids in somebody that has infection. But perhaps once the acute infection has been controlled, perhaps there is a role for that, and therefore clinical studies in that area kind of may be of value. Moving on to the next area, clinical area 2, implant-related infection. This clearly, I can see there's a lot of experts in the room on this, so there will be a number of people to pick me up on things that I mentioned. But in terms of, I'd say, the common mechanisms of failure, again, it's overwhelming sepsis. It tends to be loosening and recurrent infection are the ways that we often see the problems with the implant infection. For an infected kind of joint, we can retain the implant, the so-called DARE procedure, or we can do a 1 or a 2 stage. If it is possible to get a DARE to work, it is actually far below a mobility procedure for the patient, but the problem is it doesn't have such a high success rate as the 1 and 2 stage. So the key thing, I think, from a clinician's point of view is what can we do to try and increase the success rate of the DARE procedure? We we know it's important to operate early, but the question is how early? Is it worth doing late? What affects the success rates, and what antibiotics should be given for how long? So this is just some of the things we looked at this systematic review here. And this is looking at all the different studies that have been published. The larger the bubble, the larger the number of patients was in the cohort that was being reported. And this was the percent of success given from the mean onset of symptoms. And you can see round about the 7-day mark here, there did appear to be an inflection in the curve where the success rate kind of drops down. And that would kind of fit with our kind of growing understanding of the biofilms and perhaps when these persister cells are beginning to form. However, what was kind of interesting, I think a couple of things. One is that the overall success rate was still about 40%, even if with these patients presenting late. So that it may be that there's some value in certain patients, even in the later kind of presenting ones. And it would be good to have a way of identifying which ones of these were going to still benefit. And the other thing was this outlier group here. This one had some low virulence organisms, a high number of low virulence organisms within that, suggesting that perhaps if we could get more data in terms of the different bacteria that were causing this, that may actually kind of plug into to this. The next thing is in terms of which antibiotics to give to try and populate this. And this is some work which was done here. The bacteria have been standard Staph aureus here, being tested against a range of the common agents that we have available to us. And this is just the MIC, the standard planktonic state, showing the sensitivity. So they're all sensitive on this. But if we look at the biofilm way, this is in a biofilm model, a kind of gelatin bead model, the minimum biofilm eradication concentration. You can see that actually, for the vast majority, over 2,000, even though they're sensitive in the planktonic testing, they were resistant in the biofilm testing. And it's really primarily only this one, this antibiotic here, that seemed to have much effect, and very high dose gentamicin, which you can get with local levels, that had an effect against the biofilm. So yet we haven't done many studies that have really kind of looked at that. So in terms of the DARE procedure, ideally it should be done in less than 7 days. 40% success rate suggests that if we could identify these ones, perhaps there would be a number of other patients we could include for some later presenters. A little bit more knowledge about the organisms and how that affects things, and which antibiotics should be given them and for how long, which is a kind of a theme for quite a few of the clinical kind of conditions that we have. This is just moving on in terms of the bacterial division. This was kind of work from the Harvard group who had actually done this kind of nice thing where they'd looked at how quickly kind of bacteria were kind of becoming resistant, going from the completely sensitive state to a completely resistant state. And it was within 11 days in their kind of the kind of model they showed that the bacteria went from a completely sensitive state to resistant to 1,000 times the kind of initial kind of MIC concentration. But the clinical observation I have is that patients with chronic osteomyelitis and patients who have implant infection, it's often the same bacteria that we get at repeat procedures and repeat kind of debridements. So raising the question, do bacteria in the biofilms become resistant? And is the reduced resistance just due to the slower division rate, or else is there something else going on? And from the data from this kind of paper here, which suggests that if you look at the kind of biofilm kind of division rate, the bacteria division rate in biofilms and those in the planktonic state, it suggests that 11 days would equate to 6 months. So if we have patients on very long-term suppressive therapy, what I would be keen to know is that if we have them on kind of one for more than 6 months, could that start to tip things over into us getting kind of patients who would then start to develop resistance of the bacteria in the biofilms? Or is there a different mechanism there, which means that that won't happen even with these long suppressive treatments? So this again, in terms of my kind of little table here, recurrent infection loosening, how can we increase the success of DARE would be, I think, one of the key questions I would like to kind of more information on. And do bacteria in the biofilms develop resistance? Chronic osteomyelitis, the main area I can see where these come back, it tends to be recurrence of infection. So what is actually of interest, in the patients sometimes that we may be treating just with intermittent course of antibiotics, they have these flares of infection. But there's nothing really kind of predicts us when they get these flares and if there's anything they can do to avoid these flares. It does not appear just that when they have a kind of reduction in their immune status or anything like that, a flare happens. It just can be happen out of the blue. And I think a better understanding of what triggers these flares so that we might be able to kind of use a strategy to prevent that happening could be very useful. And the other cause, what causes our treatment failure. So I think we know that the amount of kind of bone resection is kind of critical in terms of minimizing the treatment failure. But there's a kind of conflict here. The more bone we resect, we either increase the morbidity of the procedure if we do a resect block of bone, or if we do actually kind of take out more bone and we're trying to just do a marginal resection, we can run into problems with bone resection and risk of fracture. Just checking my time now. So what would be helpful? Well, what would be helpful would be to know if we've resected enough bone, if we could actually have better ways of, during surgery, of actually detecting whether or not we've resected enough bone. At the moment, we just tend to go back until we get pinpoint bleeding. We can actually see we have vascular bone, but whether there is actually kind of an improved imaging that could actually help us to do that, I think that would be an extremely important and helpful way for treating patients. Again, chronic osteomyelitis, similar ones on the medical side, knowing which antibiotics, which route, and for how long. Unfortunately, the studies such as this one, the Aviva study, which you may be aware of, they went to the kind of massive effort of recruiting over 1,000 patients with a range of kind of septic conditions. But unfortunately, what they didn't do was really kind of distinguish with these whether or not there was a biofilm present. And what I would suggest is that whether or not you need a long period of antibiotics is actually going to depend a lot on whether or not you've left remaining biofilm after your surgical procedure. And if you've done a two-stage exchange of this, or you've done a wide excision, then hopefully we haven't left any biofilm there. And maybe they only need a very short course of antibiotics or even just kind of perioperative ones. If we've done a DARE procedure, on the other hand, we know that it's likely to be biofilm kind of still present. So the antibiotic therapies are likely to be very different. And unfortunately, the studies such as the Aviva study did not actually distinguish that. So I think there's still a big need for us to do further studies to actually evaluate what are the best antibiotic kind of programs for these patients. And it's just kind of pointing out that, in the general surgical world, they're well used to actually having this clean contaminated ones, and that perhaps we need to actually start populating ones where we look at the similar type of area, but also trying to suggest whether or not there's planktonic or biofilm still present in our patient at the end of the surgical procedure. I think there is actually a big kind of place for an increased use of local antibiotics. There's obviously a nice review here by kind of Metzmakers et al. But I think this is another area where we need to actually evaluate kind of their place further in the future. So in terms of this thing for chronic osteomyelitis, I think the main kind of failure I see is recurrence, and we need to know better how much bone to resect and which antibiotics again and for how long. Finding infected non-union, there's obviously two problems with the infected non-union. There's a failure of healing, and there's also the deep infection. So if we look at our fracture pathway, we hope that we're not going to actually have come along to this final thing where we've got non-union, infected non-union and post-traumatic osteomyelitis. So one of the things that is key is that can we identify occult infection better? We know from various studies, kind of one of our own, but others from this group here have actually also kind of shown that perhaps up to 5% of infected cases are entirely unexpected. There's no kind of clinical kind of factors that would have predicted that. So we need to have better ways of identifying these kind of patients. The other thing which I think is there is that whether or not some bacteria block the fracture repair process better than others, more than others. And that I think is an unknown area. There's nice papers that have come from these people like in terms of the mechanism of how the infection is inhibiting repair. I think these authors showed that was a prolonged kind of inflammatory phase with prolonged kind of neutrophil ones, which clearly could be contributing to it. But whether different bacteria are affecting this differently would be helpful to know. Because if we could kind of if we could override that block of infection, the block of healing that the bacteria are actually causing, then it may be possible to use a different strategy. And in terms of the strategies that we can use, I'll just come to them, just say the other thing we need to be aware of is that as we give antibiotics, then themselves may actually be inhibiting the infection. And there's a number of different useful studies here, including ones from a number of the people on stage here, which have actually shown that the antibiotics can actually inhibit the healing process. And that's, again, we need to take this more into account when we're actually treating the patients. I think we don't do this sufficiently at the moment. But coming back to what I was saying about the strategies we have for infected non-union, with the two problems, we have the deep infection and the failure of healing. There are different strategies. We can either do treat the fracture first and then the infection then the fracture, both simultaneously or neither. If it's possible to do this top procedure here first, if you can treat the fracture first and then the infection, it is actually, you can often do a far less morbid procedure for the patient than if you have to do one of the other options. So if we could overcome that block to healing that the infection is causing to the kind of the healing process, it may allow us to treat more patients with this first strategy rather than have to resort to one of the other strategies. So again, coming back to the table here, failure of fracture healing, do certain bacteria stop bone healing more, and what is the mechanism of the inhibited healing, and can we develop treatments to overcome this inhibition of healing would be questions that I would be keen to know the answer for. Final kind of slides just in closing. Clearly, in terms of the treatment for these patients that we have, there's the two components. There's the surgical component and the antibiotic component. I think we need to think further in terms of how they interrelate. In the surgery, the plastic surgeons have the well kind of known thing of a surgical ladder in terms of the different kind of procedures that they do. We have, I think, a similar kind of surgical magnitude in terms of the procedures we do. And as is there an antibiotic kind of ladder in the antibiotic treatment. For us, I think the thing that's slightly different is that the bigger procedure, it may be that we drop further down the antibiotic kind of ladder and vice versa. But again, this interrelation, I think, needs to be taken into account more in our future strategies. So my concluding slide would be this: in terms of the questions I would be keen to know the answer to. I think for the main clinical areas that I see, septic arthritis, it's ways to try and prevent that rapid cartilage degeneration. In the infected implant, it's ways to minimize the recurrent infection and to prevent the loosening. Chronic osteomyelitis, it's looking at ways to avoid recurrence. And infected non-union, how can we kind of prevent that failure of fracture healing? Thanks very much for your attention.
we have time for one quick question.
So me and my partners are often using bank powder in wounds nowadays, or Tobra, and just some of us do it just when we close. Where does that fit into your practice, and do you think that's, how does that skew some of the results? Because that's becoming more and more of a trend for those of us who are doing a lot of trauma and treating a lot of infection.
Yeah. Is that going to be covered in any of the other talks today, is it?
I don't think so.
No. So I think my understanding is the evidence for just putting the vanc powder in is not that great. There's not good evidence for supporting that, whereas there is for the use of the depot preparation of antibiotics. Having said that, do I never do it? I do sometimes do it. So I have, it's one of those things I know I shouldn't, but it's like when I have a glass of wine, too many. Actually, I do still do it. So I do put vanc powder in, but the evidence is not there for it at the moment.
So quickly that on, the Major Extremity Trauma Research Consortium is about to publish data on that, and there's kicking off a trial for orthopedic trauma in that. So that's run out of Carolinas Medical Center, if you're interested in participating. So we need to quickly move on to, thank you so much.
But I think, Josh, was it from the spinal world? The spinal studies suggested it's not beneficial.
The spine world suggests you reduce the infection by about 70 or 80%. Only the gram positives with the vancomycin. So people are starting to put tobramycin in to hit the gram negatives.
Yeah.
But there's not a lot of evidence, strong evidence, as you said, to suggest that it works. Yeah. Thank you so much. So yes. So with that, we're going to move to Dr. Chris Hernandez, who is an associate professor in the it's the aeronautical mechanical engineering and also biomedical engineering, and he's going to share the role of the microbiome on prosthetic joint infections.
Thank you so much for inviting me, Chris and Josh and Eddie, and for the opportunity to talk about our work. Let me get this running. Okay, so, okay, that's not nice. Why did it do that to me? Okay, my disclosure slide. I read this paper from Eric Paymer's group at Memorial Sloan Kettering, and they were studying hematopoietic stem cell transplantation. And there's a high rate of sepsis in these patients who undergo this procedure. And they analyzed the gut microbiome, the fecal microbiome of the patients undergoing the procedure. And this is the mortality rate that they observed. And what they saw was a big difference in the patients that had a high level of diversity of their gut microbiome. So a lot of different types of microbial organisms that are living inside the fecal material. And that actually had a clear effect on the survival of the patients after the fact. And this got me thinking, is it possible that the gut microbiome could be influencing how you get an infection, even a localized infection like we see in the musculoskeletal arena? So what is the microbiome? The microbiome is the group of microbial organisms that reside on the human body. Well, in this case, the human microbiome. The vast majority of the human microbiome is present in the gut, so the gastrointestinal system, and it consists of about 100 trillion prokaryotic organisms. There's also viruses, there's also yeasts and other microbes in there. And each of us has about 1,000 different unique species of bacteria that live inside our guts. And what we've seen in the last decade or so are an association between the gut microbiome and a number of clinical conditions, including cardiovascular disease, Alzheimer's disease, and my group has been looking at it in terms of bone disease and osteoarthritis. How could the gut microbiome influence an organ very distant from the gut? This is a cartoon where I illustrate how I think it works. Is that up here at the top is the gut with the microbes in here. Here's the endothelial cells, and here's the bloodstream. And I think what happens is the gut microbiome can influence nutrient absorption, how well we absorb nutrients, or maybe the microbes can make some vitamins that can transport it. The gut microbiome is in constant contact with the host immune system. So here's maybe a dendritic cell that's in contact. Those immune cells can then release cytokines that get distributed, or even immune cells might migrate down to the bone and joints. And the last one is just something I made up myself. I call it translocation. What I mean here is that pieces or bits of bacteria are sneaking through the endothelium and then either can get into systemic circulation. Lipopolysaccharide, for example, is all over our bloodstreams, or maybe trigger other immune responses in these regions. There might also be bacterial metabolites that are released that can have beneficial or adverse effects to the host. So this is how we think something going on in the gut could be influencing something in a bone and a joint. So I spent a lot of time learning how to manipulate the gut microbiome experimentally. I'm going to tell you about one of them because we're using the data. One is to use oral antibiotics to disrupt or change the constituents of the gut microbiome. Now, a number of groups out there that are publishing in Science and Cell will put a total of 4 different oral antibiotics in the drinking water of their mouse, and that will wipe out about 99.9% of the microbes that are in the mouse's gut. And so this is a way of sort of mimicking a condition where the mouse has no gut microbiome. What I've been doing in my lab is just giving 2 of these antibiotics, and we pick these antibiotics specifically. I mentioned this in clinicians in the room. We try and pick these antibiotics so they have a low oral bioavailability. So mostly what the antibiotics do is change the gut microbes, and they're not getting greatly distributed through the bloodstream to the bones and joints. So we're not talking about a direct effect of the antibiotics like Hamish was suggesting in the last talk, but we're talking about changes in the gut microbes themselves then having an effect down the line, okay? So you're probably not going to give these antibiotics to your patients for an infection, but it's a way of changing the constituents of the gut microbiome. Okay, as an example of how you might do this, this is a study from, that was from Japan. And what they did is they used these individual antibiotics to change the gut microbiome in the mice. They were looking at T-cell populations, and they found that one antibiotic, when they gave it to the mouse, it really changed the T-cell population. They then took the entire microbiome from that mouse, everything that was there that wasn't wiped out by the ampicillin, put it into a germ-free mouse, and then that germ-free mouse became resistant to listeria, systemic listeria infection, and resistant to tumor growth. So changing the gut microbiome can really change how the immune system is working and responding to challenges. Okay, so I'm fortunate enough to work with some great people. This is Matthias Bostrom, and in his group, in the last few years, they've developed a knee replacement for a mouse. So this is a titanium 3D printed knee replacement tibial component. It goes into the mouse's limb. I think you can see it here in this location. And you can if you're a very good surgeon, you can do the surgery. Shu Yang it does a lot of these. The mouse walks away and just fine, has a really good recovery after the fact. Okay, so it's a great way to, now Bostrom developed this to study osseointegration, but then as he was developing it, or soon after, a spectacular fellow joined the lab, Alberto Carli, and after seeing a number of infection cases, said, you know what, we need to figure out what's going on with periprosthetic joint infection. And so he led the charge to make this into an infection model. And so in this infection model, the implant is placed into the tibia, the wound is sealed up, and then an inoculation, just 2 microliters of inoculation is then put inside the synovial space to cause the infection. And in his studies, what he showed is that if he gives the right dosage of bacteria, he can get an infection every time, and he gets a biofilm on the implant. So this is what I think is a great model of periprosthetic joint infection. In evaluating, we can evaluate the infection a number of ways after the fact. We look at the gait of the animal. We can also look at radiographs, and we can identify whether or not the animal has an infection without opening it up. The other thing we do, of course, is take out the implant, sonicate it, and get a CFU count on the implant itself. We also take the surrounding tissues and get a CFU count to see what's going on there, where those bacteria have gone. One experiment we did is determine, any of you who do this kind of work, you know that inoculation really matters whether or not the animal develops a chronic infection. So we did one of these time zones. And of course, anything 10 to the thirds or higher at the implant load, we get a very high CFU count. But you get down to 10 to the second, maybe only half of the animals develop an infection, the other half fight it off. So this is the inoculation we decided to use in this study of the microbiome. And here's what we did. We had two groups of mice. One group of mice got this oral antibiotics to modify the gut microbiome starting at 4 weeks of age, and we continued until 16 weeks of age. And what we did is we stopped the antibiotics 4 days before 16 weeks of age, 4 days before surgery. Then we performed the surgery, put in the tibial component, and then added this inoculation of 10 to the second CFUs of Staph aureus Sin36. And again, the goal here was to get about 50% of the normal animals would have a chronic infection and the other half would fight it off. Here's a quick review of our results. So we had a number of components, but this is the bacterial load at the implant surface, which I think is most indicative of the presence of the periprosthetic joint infection. And on the left, you have the animals that didn't get the antibiotics. They had a normal microbiome, 50% infection rate, just like our preliminary studies. But on the right, you have the group of animals that had their gut microbiome altered by the chronic oral antibiotics, and the infection rate goes up to 73%. Why is this happening? We're not sure, but when we looked at the serum from these mice, what we saw is in the untreated animals, if they developed an infection, there was a big jump in serum amyloid A. This is a marker. It's like C-reactive protein, but it's more sensitive in mice. C-reactive protein is not as sensitive in mice. But the animals that were had the modified gut microbiome, that did not go up as much when the animals developed the infection, tended not to. We also ran some flow cytometry on cells from the spleen and from the lymph node, and we saw sort of the same pattern. The untreated mice, when they got the infection, it jumped up, but the mice with the altered microbiome, it didn't. So what we're thinking is that the altered state of the microbiome going in prior to surgery influenced the animal's ability to fight off the infection bacteria when it was present. We've been looking at the gut microbiome itself. We did a lot of quantitative assays of what microbes are there. This is what we call the taxonomics. This is telling us what species are there. And we were analyzing it. There's lots of colors here that tell us the different kinds of phyla and organisms that are there. And this is a principal coordinate chart that sort of tells us how to plot it around. And what the story here is that we didn't see much. We couldn't really tell, other than the fact that some animals had the altered microbiome and the others. So what we've done since then is do a metagenomic analysis. So instead of asking what species were there, we looked at every single bacterial gene in the fecal microbiota of the mice to try and find out what the functional capacity of the microbes there and how that was associated with the response to infection. And one thing we ended up finding in our analysis were the genes involved in the production of butyrate. Butyrate is a microbial metabolite. It's believed to be beneficial to the host, and it's a focus of many, many studies in the gut microbiome community as a way that gut microbes can influence the host beneficially. And so you see in this heat map a big difference between the untreated and the altered microbiota mice in terms of the butyrate genes. So this means the microbiome can produce butyrate. It's not transcriptomics, which will tell us if they're doing it. It's genomics, which is telling us if they can. We took out some of the fecal microbiota from our animals, and lo and behold, the butyrate content had dropped when we modified the gut microbiome. So we said, maybe this butyrate is doing something to the system. We repeated our previous experiment with a smaller sample size, and again, kept the antibiotics on. But 2 weeks before surgery, we added butyrate to their drinking water. So this is just the microbial metabolite butyrate. Did the same experiment. And these are the results. This is our preliminary data. The bacterial load at the implant surface, 50% of the untreated animals had the infection, 50% of the animals with the altered microbiome had the infection. So we think what we're seeing here is that this butyrate is rescuing that poor microbiome, or making up for the fact that the mice had a poor microbiome. So I want to thank again my collaborators, Matthias Bostrom and Alberto Carli. They're here if you want to talk to them about their models, and the rest of my team, and I have time for questions. Thank you.
Microphone to ask you a question. Chris, while they're doing that,
Yeah.
what are ways that you're going to be able to shift it back to protecting? Like what are some strategies?
Shifting the gut microbiome back
to protecting.
I mean, clearly, we're trying to figure out the mechanism, what's going on, but one way is what we suggest, this microbial metabolite that's beneficial. There may be other immunoregulatory approaches, but I guess another thing I want to point out, and this is really hot in the microbiome community, there are 5 million microbial genes in your gut microbiome, and Big Pharma sees that as 5 million treatment targets. So one day what we may have are not necessarily a drug that you would take orally that would distribute to your body. It would be a drug that would target the microbes in your gut to make them produce what's good for you, or it could just be those microbial components directly making things. So I'm excited about this because the sky's the limit, and it's really emerging right now what we can do with what the microbiome community can do. But the truth is that community, once it gets past the gut, they're mostly not interested. So when I start talking about musculoskeletal, they look at me blankly, like you would run an experiment for 16 weeks. Why would you go that long? Right? They're once it gets outside the gut, they're not as interested, and that's where I think people like us can get involved.
I could ask a quick question. So what are the direct effects of butyrate on immune cells?
I don't know the direct effect of butyrate on immune cells. So the butyrate, we know that it can have an effect direct on the endothelial cells. So the endothelial cells actually metabolize the butyrate, so it's like food for them, and that can help seal up the barrier. It can also trigger immune cells right there at the gut lining, and the butyrate does get distributed systemically. So there's just different mechanisms.
I would have to think that if it's having an effect local to the joint infection, something's got to be happening there. It can't be 100% in the gut. Something.
Well, it could be, or
this change in CFUs that you're seeing, something has to be happening locally.
yeah, I mean, it could be primary from the butyrate. It could be the butyrate influences immune cells at the gut lining, and then those immune cells then migrate or influence what's going on at the gut. But so we haven't had a chance to figure that out yet. We just have that preliminary data on the butyrate.
Have you looked at the immune response? So some of the cells, the activated, adaptive or innate cells?
All we've got so far is that beginning flow information that I showed you from the spleen and the popliteal lymph node in those animals showing a reduced response, but we don't have that for the butyrate.
Not at the fracture or the implant.
We're hoping to do that flow around the implant soon.
Okay. Well, with that, thank you.
Thanks.
So it's my distinct pleasure to welcome our next speaker to the stage, Cowie, Rudy Krishnan from the University of Rochester, and he will talk about the protection of the host immunity.
Thank you, moderators, for this wonderful opportunity to present at the first RIG session on musculoskeletal infections. Staph aureus, as most of us in the audience know, is a major pathogen in osteomyelitis, causing a variety of fracture-related infections and PJIs. What is unique about this pathogen is that it is a highly adaptable, opportunistic human pathogen. We all have Staph aureus, anti-Staph aureus antibodies in us, but are they really protective? No. And what is also really fascinating about this pathogen is that it's got a lot of virulence factors and bicomponent immunotoxins that are very specific to human receptors. Some of them are listed here. And so far, no immunotherapy against Staph aureus exists. And we have so far 17 clinical trials that have failed. And what is really appalling about these clinical trials is all of the preclinical murine model studies showed a lot of efficacy. But when it came to the human clinical trials, all of them failed. And the primary reason we believe is over-reliance on murine model systems to model human immunology. And as I had said before, Staph aureus is a highly adaptable human pathogen with very specific human immunotoxins and virulence factors. So we wanted to take a big picture approach to solving this problem. So one of the long-term goals of our research project is to try and develop a humanized mouse model of implant-associated osteomyelitis to see if we can better model Staph aureus pathogenesis and its human immune system interaction. So what is a humanized mouse? Humanized mouse are non-obese diabetic mice that are immunodeficient. They lack mature B cells, T cells, and K cells, and they also possess defective myeloid cells. And humanized mouse are generated by engrafting these NSG mice with human CD34 positive hematopoietic stem cells from cord blood. And these humanized mice have been successfully used in the fields of tumor immunology and HIV infectious disease research. And we hypothesized in this study that Staph aureus infection will be more severe in these humanized mice due to the presence of these human-specific virulence factors. And we also hypothesized that Staph aureus will indeed elicit a human immune response in these mice. So how did we test it? We utilized the well-established implant-associated osteomyelitis model in our group, and we performed these osteomyelitis infections into the NSG mice, control mice, wild-type mice, and also to the humanized mice. And in general, the humanized mice, as we had suspected, were much sicker, and they exhibited increased weight loss due to Staph aureus osteomyelitis, as indicated by the body weight change over time here. Moreover, when we looked at the bone osteolysis via micro-CT, we found that humanized mice had extensive osteolysis that was significantly higher than the control groups. And we also looked at the CFU load in the bone, and we found out that the CFU load in both the bone and the soft tissue in both humanized and the immunodeficient mice were high, but closer examination of the bacteria in the bone via histology revealed massive staphylococcal abscesses in the infected tibia of just the humanized mice. You can see here that the brown band staining of the NSG mice reveals so much more staphylococcal abscesses compared to that of the NSG and the wild-type black cyst. This was really, really exciting data that we saw for the first time. And what we also observed was that these humanized mice also exhibited increased susceptibility to osteomyelitis-induced sepsis, meaning that the CFU in the internal organs were much higher in these mice compared to that of the controls. But within the humanized mouse cohort, an interesting observation that we made was there were some mice that had very high levels of MRSA dissemination into the distal organs compared to some that had no MRSA dissemination whatsoever. So we wanted to further explore this particular interesting phenomenon to see what is really happening in these mice. So what did we do? We started with a simple correlation analysis of the extent of humanization with the level of sepsis, and we found a linear correlation of the percentage of humanization versus sepsis, meaning that the more human the mice was, the more septic it was. This was not statistically significant due to the small sample size, but it was trending significance. But what was even more exciting was that the percentage of human T cells seems to be negatively correlated with sepsis, meaning that those humanized mice that did not have enough human T cells were most septic. And this was very, very interesting for us. And we wanted to further explore the role of these human T cells in the osteomyelitis-induced septic model in humanized mice. So what did we do? We simply looked at the spatial distribution of these human T cells via immunohistochemistry and imaging in sterile humanized mice and the infected humanized mice in the infected tibia. And we found out a significant induction of human T cells near the site of sac formation, meaning that somehow there seems to be some sort of a Staph aureus mediated induction and trafficking of human T cells to the site of infection. This was really, really exciting data. So we wanted to further explore the systemic immune response in these mice. So we did flow cytometric analysis, very preliminary flow cytometric analysis of the spleen tissue to look at systemic immune responses. And here again, we found the involvement of human T cells in Staph aureus-induced sepsis. And what was interesting was the mice that had no MRSA dissemination seemed to have the maximum induction of both CD4 positive T cells that are helpers and CD8 positive T cells that are cytotoxic. So in conclusion, I would like to say here that we have demonstrated the first study to model Staph aureus osteomyelitis in the humanized mice. And as the data suggested, these humanized mice in general appeared much sicker and exhibited more susceptibility to osteomyelitis and osteomyelitis-induced sepsis. And we found this novel correlation of lack of human T cells influencing sepsis. And we believe this study, though small, is a significant step moving forward to solving the puzzle of finding a good relevant preclinical model to identify a human adapted pathogen such as Staph aureus. The study involved a lot of people. I would like to particularly thank our Rochester group, Dr. Schwartz Infection Group, and also the AO Research Institute Davos, where all of these in vivo studies were performed. And I would also like to thank the funding sources. Happy to take any questions.
We have time for a quick question or two.
Yeah. So.
How was the procedure tracking around the abscess, or you are it only when you have bone marrow?
One of the things that, these are very, very preliminary studies. One of the things that that is unique about abscess formation is that it forms a ring structure that really prevents immune cells from invading into the abscesses. So what you see here is just a representation of that. But when we quantified this particular data, but not just with one abscess that I'm showing here, but the abscesses of all of them in and around the tissue, we found out that there seems to be higher induction of T cells in and around the abscess. These are still very preliminary findings. We are still looking to see how further we can explore this trafficking. So I'll be able to answer that question better, hopefully next ORS.
Microphone 2.
I just want to applaud you. I think that this is a really amazing model.
Thank you.
I don't know if there's other instances of showing that, humanized models of infections, but I'm curious.
Yeah.
But I have said to Matthias before, you can do surgeries on mice all day long and spit in them and you will not have infection, right? And I asked him how many 1,000 mice I've done short replacement surgeries on, have any of them had spontaneous infections?
Yeah.
Right? And so that was said to us, there's something so fundamentally different. I think you're getting to the answer.
Yeah.
I create some ideas also for thinking. So one just quick question is, what cell types did you see being reconstituted all across the
Yeah, so the hematopoietic lineage was, the hematopoietic lineage, it seemed to be, the reconstitution seemed to be dominated by B cells, where typically in a humanized mouse, you see about 70% B cells and about 20 to 30% T cells. But surprisingly, you don't really see a lot of innate immune cells engrafted into these mice. So essentially, what we were told to is to looking at adaptive immune responses, but people are actually working at better engraftment technologies to make sure that we have more of the representative, innate immune cells as well. But it's a very, very nascent field, and I think there is a lot of room and scope for improvement. Yeah. Yeah, definitely. Happy to.
Thank you.
Thank you.
It is my distinct pleasure to welcome my colleague to the podium as our last speaker in the session. Jan Geurt from the University of Maastricht Medical Center is an orthopedic surgeon specializing in hip and infection treatment. And he will talk about our experience with bioactive glass, which is a novel material in the use of osteomyelitis treatment.
Yes. Thank you, Chairman, for the invitation. It's an honor to be here. And my talk will have a more clinical scope to it. I'm happy to hear that Big Pharma is interested in the microbiome because they are surely pulling out of antibiotic development at an enormous pace. So I just want to make the case for us looking into infection treatment from different perspectives as well. So I don't know if anybody has heard about bioactive glass, but basically it's been a method or a technique that has been around for quite a while. But it's actually a glass which is able to attach to living tissue, facilitate tissue growth, and bond chemically with the surrounding bone and promote new bone formation. And basically, you can play around with the composition of it with differing amounts of silicium, calcium, and so. But the bioactive glass is only a certain composition, and Bonalife, which is, let's say, the material that's on the market and available, has a 53% silicium compound and a 4% phosphate compound. There has been some rise in interest in this, let's say, technique with now dedicated journals on it, and also the rate of, let's say, papers that are published in this field rising at an enormous pace. The method itself has been around for since the 60s, and initially the applications were mainly in dental surgery with filling of defects after extraction or filling up of cysts. Also, the maxillofacial surgery side has been using this for quite a long time, filling up, let's say, defects in chronic sinusitis and mastoiditis. But in orthopedics, it's a rather new, let's say, application. And only from the beginning of the 21st century, we have started to use it in orthopedic applications. And the main interest that I had in it was in chronic osteomyelitis. So going back to the basics, it's a ceramic biomaterial, it's biocompatible, bioactive. Osteostimulative is a proven concept. It forms hydroxyapatite, inhibits bacterial growth. That's the most interesting, let's say, part of it. And it's deemed to have some angiogenetic aspects as well. And so it's important to know that S53P4 is the only bioglass on the market right now with the proven antibacterial effect. So there are a lot of bioglasses around, but this is the only one which is available with antibacterial effects. And how does this work? There is a surface reaction which releases ions from the surface of the bioactive glass, raising the pH locally and also the osmotic pressure, and this kills off the bacteria. And it precipitates onto the surface again, forming a natural hydroxyapatite, which is then of interest for the osteoblasts around, which will form bone on the surface of it. We started to use it in 2011 in Maastricht in the Netherlands. We've done about 100 cases up to date. Chris and me are involved in the clinical advisory board of Bonalife, which is also our disclosure, and I did a PhD on the subject. So what we did was we reported our first 15 patients, like I said, up to 100 now, but this is quickly to go through our first results. Starting from the beginning of 2011, we included all osteomyelitis patients. We debrided them and gave them antibiotics, and we had an average follow-up of 17.5 months. Important to know is that osteomyelitis in the past was a two-stage procedure, so you needed two operations to treat it. 11 males, 4 females, the usual locations for chronic osteomyelitis. All of these patients were operated on several times before they ended up in my department. 3 of them were hematogenous, 8 post-traumatic and 4 post-surgical, and we also had 3 non-unions in this group. Chandni classification, probably known to all of you, with, let's say, half and half, 1 and 2 stage, and the other half in the 3 and the 4 stage. And also, these were rather healthy patients, which is not always the case when you treat chronic osteomyelitis cases. We worked them up with normal lab x-rays, and we also have a lot of availability. We luckily have availability of PET-CT and MRI, and even PET MRI in the hospital. And these were the usual preoperative blood markers. So in the very beginning, we still treated them in a two-stage way with an intermittent stage with the gentamicin beads. We excised the sinus, we made a cortical window. The importance of specimens was mentioned. And we removed all necrotic tissue until you had healthy bleeding bone. Then lavage, and then we filled the whole defect with the bioactive glass. And at an average follow-up of 21 months, we had all 15 patients infection-free. Average hospitalization was 2 weeks. Antibiotic duration, 2 weeks of IV, and then about 38 days of oral, so which makes for the normal 6 weeks. Time to healing or infection eradication was 48 days. And we had a normal distribution of, let's say, the causative germs. The 3 non-unions were not consolidated at the last follow-up. This is the first patient with a chronic osteomyelitis in the distal tibia, where you can see also some small sequesters. This was one of the cases that we still did in a two-stage, so we debrided the defect and filled it up with the gentamicin beads, and then eventually filled it up with the bioglass. And interesting to see is that the granules that end up in the soft tissues absorb over time, and only the bioglass, which is in the contained defect in the bone, will form bone afterwards. This was, these results were, let's say, duplicated in larger studies. This is a multi-center study which was done in Europe in 11 centers with 116 patients with similar results. And what we found in this study was that there was a worse outcome if you had a multibacterial infection with Staph aureus and Pseudomonas. So basically, it led to a new treatment concept, in which osteomyelitis is now in essence a one-stage procedure, which obviously gives less morbidity and strain for the patient, and it's cost-effective, and I'll come to that in my next slides. And we have not seen any reported resistance to date, but the important thing is that the debridement is still the key issue. So it's not a get-out-of-jail card to just throw some bioglass in an osteomyelitic defect. The debridement is the key. Criticism of all these studies is obviously that it's short follow-up. Osteomyelitis can relapse after 5, 10, 15 years. And so we also did a cost-effectiveness study because the financial people in my hospital were looking at me, getting wary about me using this expensive biomaterial. And what we did is we set up a study in which we took a group which we treated with the bioactive glass in a one-stage fashion, 25 patients. And we took a control group from just before we started using this new biomaterial. We had no exclusion criteria other than diabetic foot, spinal osteomyelitis, and periprosthetic joint infections. So we just took the long bone osteomyelitis patients. They were all included consecutively, and we had local ethical approval. Basically, the two groups didn't differ significantly. We did have, let's say, the more serious osteomyelitis, so the Cherny 3 and 4s in both groups, which differed from the initial study that I showed. And the location was in the long bones, as I already mentioned, and microbiology was similar to, let's say, the usual causative germs in osteomyelitis studies. And so we did a cost analysis, and what we showed was that the total cost of this treatment was significantly lower than when we treated them in the normal fashion. Hospital admission accounted for more than half of the cost in each group, and we saw a significant decrease in hospitalization costs, surgical costs, and antibiotic costs, although we were, let's say, using a very expensive biomaterial, as well as imaging costs that went up. And I'll keep to time. What we showed was that we had actually a more effective way of treating these patients, and at the same time a decrease in cost. And we also did a sensitivity analysis, which gave us an incremental cost effectiveness ratio, or an ICER, which means that we save 55,000 euros for every successfully treated patient in combination with a positive clinical outcome. And the clinical result was not significantly different in this study group. But basically, we changed our treatment with these findings, and are now treating osteomyelitis in a one-stage fashion in my hospital. Thanks for your attention.
We have time for one short question, and then we go to the general discussion.
Hi, so Raj Kazan, my question is around your thoughts on, this was just osteomyelitis and maybe just chronic, right? Maybe you have some use as well, but how does this approach change in the presence of an implant? Do you see any bridge from what this does, what BioGlass does, or this indication to, or this disease state, something that involves?
Thanks for the question. We have had some, let's say, we have had some interest in looking at the application of bioactive glass in implant-related infections. My main worry, I've used it on a couple of occasions to fill, let's say, defects after behind a total hip cup implant to fill, let's say, a defect at the time of revision. My main concern with this is that if any of these particles gets into the joint, you can have devastating wear because of a third body wear. So I'm really wary of let's say, using this in implant-related infections. I think we could use it as a sort of an infection grafting technique, but then you would have to be absolutely sure that none of these particles get into the joint. So basically, the concept looks promising, but up to date, we've been very wary of using it in this indication. But I think there could be a place for it in the future. Thank you.
Thank you.
Well, I'd first and foremost like to thank our speakers. The last part of this session really is to have an open discussion on the future direction of our RIG. Open up to comments and questions from the floor, but allow me to be the one to ask the first question. Who would want to come to a musculoskeletal infection RIG next year in Long Beach? Excellent. Any other thoughts or comments on the musculoskeletal RIG in general? Right. I would ask then some chairs.
I think if you have any suggestions or topics for the future, please reach out to any one of us, and we'll try to incorporate.
Every year at the end of the ORS, you'll get a survey from the ORS. Included in that survey, I will attempt to add questions about this musculoskeletal RIG, so it'll be important for you to respond. And if there aren't any other questions, for those of you who aren't rushing out to important lunch dates, I would ask a favor to have you sit in the middle section so that we can take a photograph to archive this initial musculoskeletal infection. And with the conclusion of that, I'd like to thank you all for attending and enjoy the rest of the